All right, so the presentation is structured with an initial overview of Asimov, and then we'll talk to you a bit about the CAD-based plasmid design that we're going to implement, show some data on cell line development, and then end with how we think about using models to guide bioprocess design. Nope. Nope. Yeah.
Every single person had trouble going.
Yeah, which one is this here? Which one is it? This one on the top here?
It's this one.
Which one? That.
Yeah.
It's yeah. There you go. Awesome. So Asimov, we're a tools company, and we're based in the Boston area. So we're based in Fenway. We're 100 people, and we are a tools and services company. We're backed by the likes of Fidelity and the Canada Pension Plan. And the technology we're building really is an integration of biology and software. So on the top, I'm showing you the biology aspect, where we build GMP host cell lines. So I'll talk to you about our CHO cell line today. But I think that's the least interesting part of what Asimov does. And the more interesting part is on building genetic tools and systems. So how do we think about vectorology to optimize plasmids on a per molecule basis? We then integrate that at a deep level with various data-driven models. These models span everything from how to optimize coding sequences to how to optimize bioprocess conditions. And all of this is intermediated via our CAD platform, which we call kernel. So kernel is the way to access the genetic componentry and the models. And we're a tools company, so we don't develop our own therapeutics, but we instead partner with companies who are developing therapeutics. So there's two ways we work with companies. One is what we call the Asimov Inside model, where we can transfer the platform out to large pharma and allow them to self-serve and do cell line development. The other is biopharma services, where we do CLD and process development as a service for our partners. We've now announced over 40 partnerships across big pharma, smaller biotech, and CDMOs. And we're now working across 4 modalities. Today, I'm going to focus on biologics, but we are also developing stable cell lines for lentivirus and AAV and launching a RNA platform in the coming months. So I want to spend the bulk of the talk talking about plasmid design. I think today we've heard a lot of wonderful talks on site-specific integration and how the genomic integration is a critical part of CLD. But I'd argue that the vector design is as critical. And to just motivate the problem, I want to just talk about how the current workflow looks, where there's typically a one-size-fits-all for cell line development. A sequence comes in to the CLD group, and it can be something as simple as a mAb, or it can be much more complex. And I think we've heard a lot of questions about multispecifics. And at Asimov, we see all sorts of molecules, whether it be Fc fusion enzymes, multispecifics, and also mAbs. The problem, though, is that typically when that sequence comes in, the first thing you do is put a single peptide on it, and then you might code and optimize it with a second third-party tool, and then that entire CDS is cloned into a single platform vector. And so regardless of the molecule type, there's typically the same type of vector architecture across the board. And the challenges there are that this is a one-size-fits-all approach, which is suboptimal from both a titer and product quality perspective, but also can be high risk. If the titers are low or the product quality is poor, that can jeopardize a program or the timelines. And so at Asimov, we've thought a lot about how we can change this paradigm to a much more automated model-guided workflow. And that's what I'm showing here on this next slide, where we've kind of inverted the way we do the design, where we do the vector design, the signal peptide design, and the code and optimization in that order. But importantly, every single step is via models, and I'll show you how these work across these three steps on future slides. And the entire thing is automated, where you input a sequence into the computer, and the output is what we think are fairly optimal vectors on a per molecule basis. And what you then have is you can get, depending on the molecule architecture, you might decide to look at plasmid design space for 1 plasmid or 4 plasmids or 10 plasmids. And so I want to walk you through the type of models that are guiding this design for these various molecules. And so I'll start first with vector design. And so the first component of vector design is how do you customize the plasmid? And the answer to that is we've generated an extensive library of over 2,000 characterized components in our CHO cell line. This includes things like promoters, where on the y-axis here, I'm showing you fluorescent intensity. On the x-axis is different promoter variants. And you can see we have very strong promoters to much weaker promoters. And weaker promoters could be used, for example, to drive things with the GS gene. And we have that across the board for promoters, UTRs, insulators, polyAs, etc. So this library of components can now be mixed and matched to customize the expression vector. But the question, of course, is if you have such a complex number of components, over 2,000 components, the combinatorics explodes, right? Because you can imagine assembling these in various ways to get billions of combinations of vectors. So how do you whittle that down? And the way we do that is twofold. Number one is we have vector templates that we set based on both human intuition and also based on our previous data. So for example, we'll have a molecule template for a Morrison format bispecific, or for an IgG4 mAb, or for an IgG1, so on and so forth. That's combined with vector simulation, where we can simulate the central dogma of biology, transcription and translation, for any given vector. So what we can understand is things like what's the RNA polymerase flux going through the different transcription units. And this can be used to triage vector design and also sanity check that the vectors that are designed should behave as expected. Now, the simulation is not simulating the entire expression of a CHO cell making a protein. Right now, it is transcription and translation, but we are adding on additional modules for secretion, subcellular localization, etc., to get to a point where we actually can simulate, given a vector design, what's the end titer going to be. And I'll come back to how we do that in a future slide. So now we've been able to use models and synthetic biology to guide the vector selection given a molecule architecture. Now, this entire thing is CDS unaware. So if I have a Morrison format bispecific, you'll get the same vector templates across the board. That is also being changed over time as well. The second step is to append the signal peptides. And what we did was designed our own models to come up with novel signal peptides. And the way we did that was we developed a model, a protein language model that outperformed Signal P 6.0. And we trained that across 200 unique biologics and tested the top 1% of what we computationally mined. And what we saw is that for the top 8 signal peptides tested, shown here in blue, they outperform the published controls pretty frequently, up to a 1.5-fold increase. So what this suggests to us is that our computationally guided signal peptides are better than the state of the art. And what we can do is when we get a new CLD campaign, we can look at the predicted cleavage to choose the optimal signal peptides on a per molecule basis. So now we're at a spot where we have the vector design and also the amino acid sequence. The final step is a codon optimization. And so for that, we've developed our own proprietary algorithm, which is trained on the CHO genome statistics. And then we layer on additional biophysical constraints to further refine the model. So you have what's called a dynamic programming problem, where you're optimizing multiple variables with the same objective function. And so you get a output of the coding sequence. And the question, of course, is, does it do better than the state of the art? And the answer is a fairly resounding yes. So we compared this to what we think is the best third party. And as a kind of a side note of what we think the best third party is, we did a comprehensive comparison a few years ago of all the commercially available codon optimizers. And what we see is that in some cases, we are outperforming what we call the best third party quite substantially, up to 3-fold, 199%. In other cases, it's meaningful maybe, but much more muted, 10%, and some cases in the middle. in no case are we underperforming the best codon optimizer. And so at this point, we're at a location where we now have the entire vector sequence along with the CDS that can be cloned in, and that's molecule specific. Where we're taking all this is to go to a spot where we can do a CDS aware vector design. So as you might imagine at Asimov, we have a bunch of data on what protein sequences correlate to what titers and what designs led us there. So you can think of those as, the sequence is, in sequence space, a linear combination of letters. And then you have genetic design space, which is, thinking about like different promoters, UTRs, et cetera. We can take those and featureize them as both continuous and categorical features, and then run what's called a Bayesian optimization model. I don't have time to go into the details here, but effectively what a Bayesian optimization model does is it takes your data in and tries to train a surrogate function that represents a probability distribution of the actual data that's being represented. And then for a new case that comes in, you can then try to explore or exploit new vectors to test, and the model gets better over time. So the idea here is that for any COD campaign, we can feed that back into the algorithm, and that can make for automated vector design selection. So that can be another layer we add on along with the other tools I showed you. And so the output here, of course, is the input is a set of sequences. It's hard to read. And the output is a set of vectors that come out that we think are optimized. Now, this Bayesian optimization is right now in an alpha. We're testing this out in the lab right now internally. We hope to launch this in the next few months or so. But the idea is to use data that we're generating to then make the vector design entirely automatic and CDS aware, which kind of gets us to the end goal that we want to be at. And so that's vector design. So I hopefully have convinced you a little bit that you can take the vector design, make it very quantitative, and use models to guide that. Now, we heard a lot about integration technology. We use a transposase for integration. I think there's a lot of benefits for site-specific versus transposase. And I'm happy to debate that or have a discussion about that. But what we found is we're able to drive high copy, in some cases, 60 copies, and stable expression with very rapid recovery. So our new transposase can recover within a week or two to get a stable pool with a high titer. And the way transposase work is via a cut and paste technology where you can cut out the recognition sites and then paste back in to the various parts of the genome. So that's the vector and the integration. And so I want to just spend a minute talking about the host cell line and then kind of put it all together to give you some data on our COD workflow and kind of how all this comes together. So I think one of the most important things at Asimov is that the secret sauce is the plasmid design. We have our own host, and we have our own host because we need our own host for two reasons. Number one, for R&D, and number two, because a lot of our COD clients need a host. But importantly, all of our plasmid designs are validated across multiple hosts. Whether it's the Asimov host, a MilliporeSigma host, there's been a bunch of large pharma who've used the platform. It works in their host, etc. If the GS background or a standard puromycin background, we know it'll work out of the box. If it's a DG44, we've not tried that yet, but it's something that we think we have pretty high confidence it will work. Just quickly on our host, it's a very standard GS knockout. We got the cell line from ATCC, from ECAC, did the GS knockout and banked it at Charles River. We now have an IND in our cell line and have more coming in the next few months or so. And our timelines from transfection to RCB are very similar to what Man presented in her part, which is she had about 13 weeks. We're at the 13, 14-week mark as well. And we have stable pools within 4 weeks, 9 weeks for clone out, and then the final run is an Amber run takes us into the 14-week timeline. And we're using all the standard equipment to do all that. Now, before I go into the data on cell line development, I want to just take one step back and talk about lead optimization, because one thing we're seeing more and more with these multi-specifics is that there's a lot of ways to generate them, and there's a zoo of bispecifics that people are exploring to figure out what molecule might work well. So we're developing tools in that space to help the entire workflow. The first is around how to design these multi-specifics. So at Asimov, we're not doing CDR binding or antibody discovery. A lot of groups are using AI and other tools to do a great job of that. We think that that's well served the market right now. But we think that there's an opportunity around how you can take different mAbs and combine them into multi-specifics. And so we've developed a set of architectural exploration and computational methodologies to design various multi-specifics, where given an input of 2 MABs, you could output various types of multi-specifics. And along the way, we can scan for sequence liabilities that might impact things like expression or product quality. And so the input is MABs, and the output could be a various set of multi-specifics. Now, the question then, of course, is if you have these multi-specifics, how do they express? What's the product quality look like? And you can, of course, test that in a transient system. That's what's done right now. But we've developed rapid stable pools that allow you to test this in a stable format. And so this can be from the input we give, or comes from a partner themselves, where you have a candidate of n molecules, n could be 50, 100, whatever it might be, that's your final lead candidates you want. They could be tested in transient, but that transient data doesn't correlate well with stable data. And so what we've developed is a high throughput way to develop stable pools. This can be up to hundreds of stable pools, and those stable pools are very correlated with the final clones that come out the back end. And so in a 48-well plate, we can generate these stable pools, do multiple plates, and get a titer and PQ readout, where then you can rank order your list in terms of titer, but probably more importantly, the key product quality attributes, whether it's a high molecular weight species, whether it be the percent of the proper heterodimer, et cetera, et cetera. So we think these tools in the lead identification regime are very complementary to our cell line development workflows. And so that's a bit on CAD-based plasmid design. It's a lot to throw at you, interspersed some data, but kind of philosophically how we think at Asimov about CAD-based plasmid design. So moving on to how this translates to the real world. So first of all, I guess it's a fair question to ask is, does vector design exploration matter, right? Like maybe one size fits all is good enough. And at least from our experience, the answer is a resounding no. So here's an experiment we did a few years ago where we took 4 molecules, all 2-chain, across 6 vector variants. And those 6 vector variants explored things like heavy chain light chain ratio, the GS selection stringency. And what we find is that there's not a singular vector that's good across all 4 molecules. So if we chosen vector 1 as our platform vector, it would have been great for this MAB, but would have been suboptimal for 3 of the 4. And the same thing is true kind of across the board. And this really does motivate the need for vector-based design. So in our CLD workflows, the plasmids come out the back end, and then we can test and make stable pools for multiple plasmid variants, typically 6, sometimes 12 variants. We then advance the top one to clone out because we see a very good correlation between the pool titer and the clonal titer. And what we see is, across the board, 8 to 11 grams per liter before any process development. I know it's really hard to read here, but the top 3 are bispecific antibodies, a 3-chain, a 4-chain, and a 2-chain. Sorry, it's out of order. And then a set of MABs as well. And kind of across the board, we're seeing strong titers. But I think that titer is only one dimension for cell line development, right? We talked about product quality quite a bit. And we think that the power of vector design is not just for searching to optimize titer, but to optimize product quality. So here's a case study for this 3-chain bispecific, where during the COD workflow, we didn't just measure titer here on the x-axis, but we also measured the heterodimer percentage. And if we just chosen to clone out the pool based on the titer, we would have chosen one with high titer but very poor product quality. And instead, we chose one that was decent titer but really good product quality. And we do the clone out, we're able to get over 11 grams per liter and over a 80% heterodimer. So this again shows the need for searching vector design space to optimize across various attributes. And I'll leave the molecular biology section with one final case study. This is a little bit more of an esoteric one, but I think pretty interesting, where it was with a large pharma partner, and they came to us with a hard to express bispecific, this is a Morrison format. But interestingly, they had 3 binders that worked really well and one that was very poor. And they'd thrown a lot of resources at it. They were open to amino acid mutations, but they asked us, can you come help us fix this? this was a little bit of a, an atypical workflow because of how much they've done at this. And so what we did was a kind of a deep multi-omics debugging where we looked at transcriptomic data and also translatomic data, not shown here, to identify what's the defect going on. And what we found were two things. The first was that there was aberrant splicing in their initial vector they designed. And the second was a ribosome was actually stalling during translation, causing nonsense-mediated decay. So we made an amino acid mutation to solve that second problem, and then did a new codon optimization to solve the first problem, and we were able to improve the titer almost 70-fold, and actually beat the titer of 1, 2, and 3. And we're actually publishing this data with this large pharma partner. That publication hopefully is forthcoming in the next few months or so. So with that, I'm going to step on to the final section of the talk, which is around how do we think about bioprocess design? So I've told you a lot about how we think about what goes into the cell, but how do we think about what goes around the cell, the bioprocess design? And so to that end, we've developed an entire set of models here that can allow us to predict, given the process variables and the metabolites, what's the end titer going to be? And so what I'm showing you here, and I know it's a little hard to see, is it's traces of our training. The little dots are all training actual data, and the curves are the fits we get from our model. And this model is a hybrid mechanistic and machine learning model to capture the cell state during the course of metabolism, the bioreactor run. And so we trained this model extensively across a lot of amber runs, flex data, et cetera, across different molecules. And just to give you a sense of the breadth of data we're dealing with, these are the type of fits we're looking at. I don't expect you to take much away from this, except that it's a ton of data and the fits look pretty good. And so we can then challenge the model to then ask, can we identify various conditions that are supposed to improve the titer? And here, a lot of these on the top are metabolites, but the one in the bottom here is a protein titer. And maybe you can see it, maybe you can't, is in teal here is the original predicted titer, and in green is the model guided titer. And using this, we can test those parameters out, and we've seen up to a 20% gain via just model guided process development, clone specific. So real gains we're getting from our initial data around bioprocess optimization. But it doesn't just end there, right? You can imagine using this on a clone specific basis, but you also can think about using this to improve the COD platform process. So the way we think about that is, right now, our process before was to screen 144 clones. The top 48 advance to an amber, and then they're run in a 48-way amber with a singular platform feed. We've developed some cool algorithms where we can, at the 144 clone stage, sample, kind of identify which ones we think will be the winners. So whittle the 46 down to 16. Now that frees up a lot of room in the amber system, the 48-way amber. So you can now test out 3 different media feed combinations, and those media feed combos are model guided. And here's the data around that. So here's kind of the feed regime around how these are pretty complex feeding strategies for both strategy A and B that were predicted by the model. But here's the data. That graph, I'll try to walk you through it. On the y-axis is the percent gain over the platform process. And there is the feed strategy A, and then feed strategy B, and then kind of the maximum of A or B. And what's super interesting is that there's always a feed strategy A or B is always better than the platform feed strategy, but it's not always A or always B. So for example, in clone 1, strategy A was better. In clone 4, strategy B was better. If we'd only tested feed strategy A, we left titer on the table. Only feed strategy B, same story. But because we're able to do kind of a mini process development in the amber doing cell line development, we're able to kind of get the best of all worlds. So I want to just leave you with kind of how I think about where we're going with all this, the kind of the roadmap for what we're trying to build, which is what I hope is a truly in silico guided COD and PD workflow. I'm hoping you see kind of the bits and pieces getting there. But if you think about how things are done today, it's fairly empirical with the vector cloning, cell line development, media development, and the process design. And if something goes wrong in any of those stages, there's got to be an iteration cycle. And our approach is really to make the entire thing in silico driven with experimental data to drive those models. And so the genetic design that we put is able to predict what's the optimal cell line development path, the optimal process path, and the optimal media conditions. Now, again, this is a roadmap that'll take years to get to, but I think we're making substantial progress towards that. And that's the vision I'm excited by for bioprocessing, which is to make the entire thing, model guided. So with that, I'll open up for questions. And, thank you so much, guys. We do have a booth here. I don't know the number, but, please come visit us.
Thank you, Raja. Nice presentation. So do people have questions?
Hi, I have a question mostly on that last portion.
Yeah.
For the studies where you were looking at metabolism and looking at feed rates, were you using a proprietary feed or were you using off the
These are all
shelf?
off the shelf. It's just the amount and the feeding schedules varying. But we are looking now to make our own proprietary medias and feeds. So that's kind of the next step. But right now, it's all off the shelf.
Thank you.
So you mentioned that you used models and machine learning to narrow down your clones from 144 to 16 or 18.
Yeah.
Could you talk about how that is done and what data are you inputting into the models? And
That one, I'm probably going to defer on that question and maybe get back to you afterwards. That's okay. On what I can disclose there or not.
All right. Okay.
Yeah.
Thanks.
Hi, I loved your talk.
Thank you.
My question is regarding the input of the vectors that you're adding for training. So is it a standard vector design that you input? Because I'm assuming if this is for industry, customers do tend to have a bunch of different methodologies by which they want to upload. So do you have the capacity to build an API to parse that? And
you mean for if you had specific data for that we want to upload for your specific? Yeah. So the way we do it, at least for ourselves, is you can featureize in multiple ways. So sequences are featureized, kind of continuously, but the vector design, you can code what kind of promoter it is, what UTR it is, what's the orientation. So you kind of get a categorical feature and you kind of code it that way and upload that. So I don't see any reason why that just could not be applied one-to-one to your data over there.
Okay. Yeah. Thank you.
I don't know if I answered your question or not. Was that your question?
Yeah, somewhat.
But happy to chat afterwards as well.
Sure. Thank you.
Very good titer at 8 and 11 gram per liter. I'm curious if you got the same titer for biospecific and multispecific.
Yeah, so it was really hard to read in the gray text box, but the top 3 boxes were actually multispecifics. One was a 2-chain bispecific, one a 3-chain, one a 4-chain, and all of them were in the 8 to 10 gram per liter regime. So yes, and those are high product quality. So it's all fairly good protein coming out the back end, not just a bunch of gunk.
That's very good. And when it comes to how to express protein, normally the challenge comes that the impurity goes 50%, 60%, making hard to produce a certain protein.
Yeah.
Did you have insight that you have completely resolved this issue, or is it something still need a lot of development?
I think the word complete is an overloaded term, so I will never say everything. There's always a tail end problem you have. I think that vector design can solve, where balancing chain ratios, for example, or you want to slow down translation time to allow for the protein to fold properly. All of those are within our wheelbox to solve. But we have had just once in a while a molecule that's so intractable, it's just a molecule problem. And that's actually why then the developability stuff that we're working on is kind of a complementary toolkit where if it's such a bad molecule, are you open to then change the amino acid sequence, to make it, more expressible? So I would not say everything's solved. I think that if we solved 80%, I'd be very happy. And, I'll push you to get to 90 or 95, but I think there's always gonna be a tail that you can't solve.
Thank you.
Thank you. So there's one slide you have a model that predict titer with some metabolized input. I wonder if those are from just NovaFlex or there are additional metabolized data you're inputting to the model.
Yeah, it's a great question. So the training data is NovaFlex and then also amino acid data, so essential amino acids. And then also bioprocess conditions like DO, pH, things like that. Yeah.
Okay. A few other ones.
Hi, I'd like to follow up on a question the lady over there asked before, and that's with, of course, every company has their own flavor of platform, especially when it comes to multi-specifics. So in my former life at a big pharma, I spent a lot of time looking at dozens and dozens of different multi-specific platforms, and they're very different. There's a lot more variety than just changing your two binders. And also you may or may not have insight into what that is. So how do you make this more generalizable? Because I think it's a great tool, but it's got to fit a lot of very, very different, structurally
for
different
sure.
platforms.
Are you talking about the multi-specific, like the part where you put the mabs going to the
like there are a lot
of architecture design?
Huge variety of
100%
architectures.
agree. And I think that this is just like our initial foray into multi-specific design. As you pointed out, there's so many different, variations. You want getting the RF mutation, knob and Hall, blah, blah, blah. So there's a lot of stuff that you have to think about when you design the multi-specifics. We're kind of trying to get our feet wet right now. To be honest, this is a newer offering for us, which is designing multi-specifics from, two binders. But I think you're absolutely right, which is that you're not going to be able to solve all the problems. And I think that one thing that we're seeing more and more is that various companies call us on different architectures for whatever reason. And so I think we're trying to take that data in and kind of improve the offering, but it's kind of a more nascent thing we're launching right now, trying to understand how can we help with the design, just because we're seeing people come with really, Frankenstein molecules that we think could be better if they were designed a bit better.
That's exactly what we used to call them, Frankenstein
molecules.
Exactly.
And I was a protein engineer in my previous life, computational. So I think that there's a lot of, a lot of good stuff we can do there, especially with these new models coming out. But this is just, our first foray into it. So.
Well, I think it's great. And also these are a lot more challenging in terms of manufacturability. And then they're hellishly often sticky and just not well behaved.
Exactly.
So if you can optimize that, that would be fabulous.
And what's super cool is that we've shown also, and others have shown too, that you can do these protein simulations and molecular dynamic simulations. And those can predict things like expressibility, aggregation, et cetera. So it comes into what we can predict computationally. And it's not that you're going to solve everything computationally. It's just that it helps you whittle the space down on what you might search. And then you can just make, rapid pulls from them and then go from there.
Great. Thank you.
Yep.
Really nice work.
Thank you.
Excited how you guys are bringing everything together.
Inspired a lot by a lot of your work. So,
So I was curious about the modeling part at the end. This hybrid model that you're looking at. First thing, how many runs do you feel like you've needed to be able to start training it? And is this all fed batch? Do you have perfusion?
My understanding for the model training, and I'm not the expert here, is that it's about a couple dozen runs to train the model of AMBER runs, flex data, amino acid data, et cetera, to get the initial model calibrated. This is all fed batch AMBER 15 that we've trained on, a bit of 250 data. We've not done perfusion, something I'm pretty excited about, but it's just not been. I feel like perfusion goes in ways of people doing it than not doing it. I think maybe we're going back in that curve right now. And I'd love to build models for perfusion as well, but all this is fed batch at the moment.
Great.
Okay, well, thanks for the presentation. I think we went through all questions.